Welcome to Oxidative Phosphorylation. So today we'll really wrap up our understanding of how ATP is synthesized in oxidative metabolism, and we will finally understand why high energy molecules like NADH and FADH2 are considered high energy. Here are our learning outcomes for the day. So, as just mentioned, we'll finally understand high energy compounds and why they're considered high energy. We'll be able to draw and describe the various subunits of the ATP synthase, in particular the alpha-3 beta-3 subunit, the gamma, the C ring, and the A. We will be able to draw and describe the structure-function relationship of some various subunits. And we'll predict whether ATP will be synthesized if inhibitors are used on components of the electron transport chain, or if proton couplers are used, again, seeking to understand flux through these pathways and eventually connect glycolysis to the TCA, to the ETC, and to ATP synthesis. Our Meet a Scientist moment today is Holden Thorpe. He is the editor-in-chief of the Science journals. Science is a major publication of all science types of research in the United States. He is autistic. He is on the autistic spectrum. And he studied electron transfer reactions of nucleic acids, which is some pretty interesting stuff that we won't get into for Biochem 1 or for comprehensive biochemistry, but still cool. So, let's think about what we have learned so far and try to make some connections here. Last time we were together, we saw how electrons moving through the electron transport chain, either from NADH or through FADH2 or through glycolysis, NADH, end up moving through the electron transport chain. The movement of these electrons results in protons being pumped across the membrane. At this point in time, folks, we can see how we have been pumping all of these protons into the inner membrane space, and this results in a high concentration of protons in the inner membrane space. If energy is released by the movement of these electrons through the electron transport chain, we can understand that that energy release is stored as the proton gradient, because the energy release was used to move protons from a low concentration to a high concentration, which we would say is against their gradient. Today we're going to see that these protons were stored, the energy was stored, and it's going to be used as they fall back down their gradient to eventually synthesize ATP. There are some key observations about ATP synthesis that are going to be important to note, because these kind of informed our understanding of the structure. So some scientists saw that oxidative phosphorylation requires an intact inner mitochondrial membrane. They noticed that the inner mitochondrial membrane is impermeable to ions, so no ions cross via diffusion. 3, they noticed that there is a measurable proton gradient from the matrix to the inner membrane space. And they noticed that small molecules called uncouplers permeabilize the inner membrane. And when this happens, the electron transport chain works fine, but ATP synthesis stops. So these are some key observations that informed what we know about ATP synthase today. What else do we know? We know that the synthesis of ATP under physiological conditions generally costs 40 to 50 kilojoules per mole. We know that each proton falling down its electrochemical gradient provides about 20 kilojoules per mole. All in all, we estimate that it takes about 3 protons falling down the gradient to synthesize a single molecule of ATP. The enzyme complex that performs the synthesis of ATP is called ATP synthase, or sometimes complex 5. It is super cool because it couples physical movement to a chemical reaction. Today, we're here to see how it uses the proton gradient to synthesize ATP. On the right-hand side of the screen, folks, is an electron micrograph of the ATP synthase in mitochondrial membranes. Each of these things that you see here that kind of looks like a lollipop with a little stem and a ball is an ATP synthase. It has 2 major parts. The F0 is a transmembrane unit comprised of roughly 8 individual subunits. The F1 is a peripheral membrane protein comprised of about 5 individual subunits. Let's walk through each of these together. Up next, we'll learn about the F0 subunit.